嗯，好，大家早安、下午好以及晚安。我知道这个就是有很多人从不同的 time zone 或者是不同的城市 join 这样，然后因为我还是习惯就是 present 我的 research in English，so I will talk about my research in English, but feel free to use your comfortable language if you have any questions. Okay. So yeah, it's very nice to meet you all here and thank Frank for inviting me to share my research. Um, Today I'm gonna talk about my graduate studies and my current research, which focus on effects of locus coeruleus and aging on decision making. So this is an outline for my talk today. First, I will talk about locus coeruleus, its association with cognitive function, and the assessment I use in my research. Sorry, this is something. Okay. Then I will share two of my graduate studies, which focused on the relationship between aging and the locus coeruleus norepinephrine functioning in decision making. Lastly, I would like to briefly talk about the concepts of my current ongoing study, in which I'm trying to understand the association between aging and the locus coeruleus norepinephrine system on intrinsic motivation. Okay. So locus coeruleus, as can you see my mouse here? Like, or
Yeah,
do
yeah,
okay.
we see your mouse. Yeah.
Okay, great. All right. So the locus coeruleus, like this blue dot, like a spot, which in Latin means like a blue spot because it's colored in blue in postmodern sample. It's a very tiny nucleus, just like a piece of rice. So it's like located in the posterior pons. Although it's tiny, locus coeruleus is the main source of norepinephrine neurons, and it regulates the arousal and autonomic functions associated with the frontal and limbic networks. The other hand, locus coeruleus is also one of the first regions to accumulate abnormal tau protein in the brain, which is associated with like Alzheimer's disease. So nowadays, there are some approaches to investigate like locus coeruleus, for example, postmodern samples, animal models, or neuroimaging assessments. So because I do human research, most of the time we use neuroimaging techniques to assess the locus coeruleus functioning. So first, I would like to introduce the structural MRI. So nowadays, there are some spatial sequences, for example, magnetization transfer, turbo spin echo or multi-echo fast low angle shot. These like sequences can like assess the locus coeruleus and also visualize it. But why do these sequences work? It's because of a neuromelanin. Okay, so neuromelanin is the byproduct of norepinephrine stored in the locus coeruleus that has a fruitful iron and copper to show T1 shortening effects and further resulting high signal intensity on the image. For example, here, as you can see, very like bright two dots, they on this MR image, like it's the bilateral locus coeruleus. So we usually also use the protein reference area as the reference, because it doesn't have a neuromelanin. Then what we usually do is we compare the signal intensity in the locus coeruleus masked region with the signal intensity in the protein reference area. Okay, to compute these like locus coeruleus MRI ratio to indicate locus coeruleus structural integrity. Okay. So in aging research, one recent study recruited the participants aged from 20s to 80s across the lifespan. So, and they also use this type of MRI to assess locus coeruleus structural integrity. They found that they found a quadratic relationship between locus coeruleus structure and age. The peak occurred at the age around 60. Okay, so the increase in teenager and young adults is related to the accumulation of neuromelanin in the norepinephrine neurons, and the decrease in the old age is due to the neural loss. Okay. So if we and then previous study also found, especially in the rostral locus coeruleus, positively correlated with memory performance or attentional shifting ability. It's because if we go back to the anatomical pathway, as you can see here, the rostral LC projects to the cortical cortex, while the caudal LC connects to the spinal cord. On the other hand, some studies also showed the association between locus coeruleus structure and Alzheimer's pathology. So lower locus coeruleus structure is associated with higher abnormal tau accumulation in the brain. Another neuroimaging technique we also use in our lab is the PET. Okay, especially we use these fluoro and tyrosine or FMT tracer. This tracer is a substrate for aromatic amino acid decouplase. Every time I say this word, is like I'm rapping. Okay, so or AAAD. This tracer is very sensitive to catecholamine, including norepinephrine and dopamine or serotonin synthesis capacity. Okay. I know it's difficult to understand the neurochemical mechanisms by looking at the text here, or it's like a chemical formula here. So I prepare a cartoon example. So let's imagine that our brain is like a factory which uses different enzymes to change one neurotransmitter into another. Okay, so the origin is like a L-tyrosine. With this enzyme, it changes to L-dopa, and then with the AAD, it changes to dopamine, then norepinephrine. So our FMT tracer is to target this, our replacement of the AAD here. Okay. In our previous work, use this FMT PET tracer, we found an interaction between norepinephrine synthesis capacity and tau pathology on memory performance in older adults, especially like low and medium synthesis capacity, the orange and red line here, was associated with high abnormal tau accumulation and also worse memory performance. In addition, in my earlier work last year, we further found that the interaction between locus coeruleus MRI structure and locus coeruleus FNT PET signal predicted cognitive function. So in this study, we had a bunch of neuroimaging data and also a battery of cognitive performance tests. We use a partial least square correlation to extract latent variables on both sides that explains the most variance in these two data sets. So first, we found a correlation between the latent locus coeruleus brain and latent neuropsychological performance, which means people who had a higher locus coeruleus brain functioning showed better cognitive performance. Especially the rostral especially the rostral and medial locus coeruleus. Earlier I showed you this part projects to the cortical cortex regions are the primary contributors in these like lantern variable. And these primary contributors were correlated with the neuropsychological performance related to processing speed, cognitive flexibility, and cognitive stability, like more related to executive function. So far I have shown you the measurements of the locus coeruleus in the brain. Another psychophysiological assessment I use in my study is pupil dilation. So pupil dilation may serve as a potential window of the locus coeruleus activity. Why? Is because there are many connections between pupils and brain regions. I know it's very complex, so I'm gonna like focus on key information and guide you like through one by one. Okay. So first, let's talk about the peripheral nerve system. We all know the light refraction, right? So bright light results in pupil constriction, and dim light causes pupil dilation. These two pupillary responses are modulated by parasympathetic and sympathetic nerve systems. The parasympathetic system is like a default system. So when people at rest, the parasympathetic system controls the iris muscle to constrict pupils. In contrast, when people get alerted or engage awareness, for example, during cognitive tasks or listening to my talk right now, the sympathetic nerve inhibits the parasympathetic pupil constriction, which results in pupil dilation. Now we move one step back to the brainstem region. So in the brainstem, this one commander is called Edinger-Westphal nucleus. So this nucleus is belonged to the parasympathetic system to constrict pupil because the locus coeruleus has a direct inhibitory projection to Edinger-Westphal nucleus. When locus coeruleus release norepinephrine, it inhibits the Edinger-Westphal nucleus from constricting pupil muscle, resulting in pupil dilation. Okay. So in the previous animal models, they already show like the positive association between pupil dilation and the firing activity in the locus coeruleus neurons. In humans, pupil dilation is also related to phasic locus coeruleus activation during cognitive tasks. So now I have talked about locus coeruleus structure and its neurochemical function and the association with pupil dilation. So my research mainly uses these measurements to investigate to what extent edge may modulate the locus coeruleus norepinephrine system and affect cognitive function. So in one of my graduate studies, I studied the effects of adult age and locus coeruleus norepinephrine functioning on reinforcement learning. Okay. So what is reinforcement learning? In our daily life, we make a lot of decisions, right? So some of our decisions are made based on previous experiences, and this is an example. So, every time I go back to Taiwan, I'm so confused by some fancy drinks name. So, for example, the like 狸猫贵妃, if we translate it in English directly, is noble lady, like a civet cat, or 黄金熊猫, the yellow crystal pandas. I have no idea what like these drinks are. So just saying that it's not an advertisement, so just an example. Okay, so because I have no experience with both drinks, my predicted value to these drinks are equal. Okay. So if I choose the panda one, and then it has the good, then because of the good experience, I increase the value of panda, or vice versa. If the drink is bad, I decrease the value. Okay. So the value of your chosen option is updated by the outcome or the reward prediction error in a precise way. So the reward prediction error is the difference between your actual outcome and your predicted value. So we can use the Q-learning model here to indicate this learning. So the Q stands for quality, which indicates how useful a given action is to get future reward. If an action has a high quality, then people tend to exploit the option. But if the option has a low quality, you may explore to another choices. So when make a decision, we need to do value computation, comparison, and updating. So these valuation processes are associated with nucleus accumbens and medial prefrontal cortex underlying the dopamine and norepinephrine systems. So when aging attenuates dopamine and norepinephrine systems, it presumably results in impairments in the value representations and updating, but it's not clear to what extent the age-related decline in the locus coeruleus norepinephrine functioning in humans may affect decision-making and reward learning. So in this study, I conducted a probabilistic decision-making task that involved different reward probabilities. So in this task, there were 3 choice pairs with different reward probabilities. For example, 80%, 20%, 70-30, and 60-40. So this would be the most difficult one. Participants didn't know the probability knowledge at the beginning, so they learned it from previous feedback, which indicates whether their choice gained or lost 5 cents. Then I applied a Q-learning model here. So in the Q-learning model, there is a reward prediction error, indicate the difference between the actual reward and your expected value. The Q value is updated by this reward prediction error, but is when by the learning rate. Okay. The 2 learning rates here in the Q learning model do not really indicate how good people choose the higher rewarded options, for example, 80, 70, and 60. But the 2 learning rates indicate the degree to which the reward prediction error is incorporated in the Q value. So if the learning rate is high, which means the Q value is more based on the recent reward prediction error. Then lastly, participants' action is determined by a soft mass function, which indicates how likely you will choose this option. So there is a beta parameter here. The beta parameter indicates a value sensitivity. Higher beta value indicates greater sensitivity to higher rewarded options and more exploitative to a choice. Of course, I measured participants' pupil dilations during the task as a psychophysiological toolbox and focus on the choice and feedback phase. Okay. In this study, we recruited 42 younger adults and 41 older adults. Lastly, we also assessed the locus coeruleus MRI contrast as an index of the locus coeruleus structural integrity. The red area here indicates the locus coeruleus region, and the blue area here indicates the protein reference area. According to previous literature, we compare the signal intensity in the locus coeruleus red region by comparing the maximal signal intensity in the locus coeruleus muscular region with that in the protein reference area. Just unfortunately, in this study, because of COVID, we were able to scan only like 25 younger and 34 older adults. Okay. So in the results, first, compared to younger adults, older adults overall showed lower accuracy in selecting the higher rewarded options. Older adults also show lower differences of value beliefs, the Q values. And this effect was mainly present in the choice pair with the smallest difference between the probability ratio, which means the most difficult one, the 60-40 pair. And another parameter in the Q value model is the beta. We found a higher beta parameter value in younger adults compared to older adults. So older adults, they show like a lower value sensitivity. For the pupillometry results, in the choice phase, we found younger adults' pupil dilation at each time point negatively correlated with the Q value difference between the two paired choice options. So, which means lower value difference, like a greater lesion uncertainty, it would be like the 60-40 this pair, elicited larger pupil dilation. And these like uncertainty effects, so these negative correlation indicate the uncertainty effects in the pupil dilation. In older adults, although older adults also show this negative association, the effect was smaller and only appeared later. The black line here indicates the significant edge differences. So the smaller and reduced effects in older adults may indicate a noisy neural information process of the various representations and comparison. In the feedback phase here, again, younger adults, so in the feedback, participants had to update their value belief or their Q value according to the outcome, the feedback that they get right now. So again, younger adults' pupil dilation negatively correlated with the Q value of the chosen option, which is still like indicates the uncertainty to the reward probability from the feedback. And this effect was only observed in younger adults, but not in older adults, which might indicate age-related impairment in the value estimation and updating from feedback. For the MRI part, we found older adults showed lower locus coeruleus MRI contrast compared to younger adults, which may suggest age-related declines in locus coeruleus norepinephrine functioning. We further found that older adults who showed a higher locus coeruleus MRI contrast, which might indicate better norepinephrine functioning, showed better value sensitivity to exploit higher rewarded options. So in this study, we found a reduced uncertainty evoked pupil dilation in older adults, suggesting impairments in value computation and updating during reward-based learning. Lower locus coeruleus MRI contrast in older adults may indicate reduced locus coeruleus functioning and maintenance of the norepinephrine functioning in old age may benefit reward-based decision-making and learning. Okay. So far, do you have any questions?
如果就是因为第一个 study 已经结束了，所以如果大家有问题，可以趁这时候就先问一下，然后不然就是讲者可能就要进入他第二个研究 section 这样子。
Yeah.
对，
Or if you need some time to kind of wrap your question, we can also like have the question and session at the end. Yeah. Okay. All right. So in study one, participants didn't see the reward probabilities in the choice options, and then older adults showed impairments in various presentations and updating. If we show participants, especially older adults, we show them the reward probabilities in text. Would older adults still show reduced sensitivity to various representations during decision making? If it's still the case, is there any way to increase their value representations or to improve their decision making? So this is what I did in my study too. So in this study was a collaborative project with Professor Josh Gore at National Taiwan University. So I used their like lottery task. In this task, we show participants a lottery trial by trial. So in the lottery, we show them the probability of winning, probability of losing, and the magnitude of the reward that they would get. So participants had to decide whether they want to accept this lottery, this gamble, or not. If they accept, there is a probability to get this amount of the reward. So we show them the outcome of the current trial and the accumulated reward. If they reject this lottery, they didn't gain or lost any point. Okay. So in this study, we recruited 44 younger and 51 older adults. We still found age differences in the choice performance, especially older adults. They tended to accept more gambles when the probability of winning is low, and rejected more gambles when the probability of winning is high. So older adults still show reduced value representations even they were presented the reward probabilities. Now the second question is, we try to know whether there is any way to improve older adults' decision making and what's the mechanism behind. In this study, we added another condition, which uses a color-coding scheme as a decision aid. So green indicates the probability of winning and red indicates the probability of losing. The procedure is the same as the control condition here without the color. So participants had to decide whether they want to accept this gamble or not. So to compare the color coding scheme addition add with a control condition, I fit the choice behaviors into a logistic regression model with a continuous variable along the expected value. So the expected value is computed by this formula. It's probability of winning times the magnitude minus the probability of losing times the magnitude. Okay. We focus on the slope of the logistic regression, which may indicate the value sensitivity. So overall, younger adults showed steeper slopes than older adults, which may reflect a higher value sensitivity than older adults. The decision aid, the color coding scheme, in the gray lines here, increased the slope in both age groups. Okay, so which means like the green and red color coding, like a decision aid, did improve participants' decision making. We also measure their pupillometry data during the task, and for the results, the decision aid, the gray lines here, decreased the pupil dilation in both age groups, and the reduced pupil dilation may indicate a reduced cognitive demands when computing value representations. Okay. So in this study, we still found less distinctive representations during value-based decision-making in old age, which may be due to impairments or like deficient neuromodulation with noise information processing. If we increase the information saliency during the decision making, the catecholamine skin would reduce the cognitive demands of value representations and like computation and comparison. So to make the value representations of positive and negative outcomes more distinct. So older adults, they are like a benefit from it to improve their decision making. So with these two studies, oh, by the way, do you have any questions for this study? If not, then I will continue to my last study. My current ongoing study. Okay. So with these two studies, I consistently found age-related declines in decision making with intrinsic value involved. So which is the monetary reward. Okay. So after I finished my thesis in like Germany, one day I was sitting by the Elbe River in Dresden. By the way, this is like a true advertisement now. So if you have a chance, I highly recommend to visit Dresden. It's a very beautiful city. Okay. So I was sitting by the Elbe River. I started thinking, can people's decision-making be affected by intrinsic motivation, for example, curiosity? And to what extent curiosity-driven decision-making is associated with locus or norepinephrine functioning? And how does age change curiosity-driven decision-making? So I developed a task to address these questions. This is my currently ongoing study. Before I started the current study, I looked into previous literature and found that some studies propose that locus stores is the critical drivers of intrinsic motivation. Especially in younger adults, they also found curiosity modulates pupil dilation and enhances memory using the trivial paradigm. In animal models, they found lesions in the locus coeruleus reduce the exploration of novel environments. And also cognitively normal older adults showed a reduced curiosity trait compared to younger adults. And this association with Alzheimer's disease. So Alzheimer's disease patients showed reduced novelty seeking, but these are pieces of evidence. So in my current study, I try to use a multimodal approach, including pupillometry, MRI, and PET, to investigate the effects of aging and curiosity in decision-making and memory, and the association with locus stores and norepinephrine functioning and tau pathology. So in this study, we will aim to recruit 60 younger and 60 cognitively normal older adults. And I developed a task which is called photographic art and storytelling study. So in this task, we show participants some photos and we tell them each photo has a story behind it. We ask the participant how curious you are to learn the story behind each individual photo and your like opinion after reading a story. Okay, so this task uses different levels of self-reported curiosity to determine the effects of curiosity on decision making and memory performance. Also, I will measure their pupillometry during the task. and the MRI data focus on the locus coeruleus structure and resting state functional MRI. Because we will recruit older adults who already participated in my mentor's currently ongoing NIA study. So we will have the PET data related to tau accumulation and norepinephrine synthesis capacity in the brain. Then the first aim in this study is try to determine the effects of older age on curiosity-driven decision-making and memory. Next, we want to define the association between locus coeruleus structural integrity and curiosity-driven pupil dilation, and then their conjoint role in predicting memory. Because of time and we are still doing data collection, so I'm not able to present the details of this study and the results to you now, but I will have a poster presentation in CNS Cognitive Neuroscience Society conference this year. I'm hoping to have a preliminary results by that time. So if you will attend and if you are interested, please stop by my poster. Okay. So just in general, in my studies, we found attributed decline in information processing of intrinsic values during decision making. In my current study, I try to leverage curiosity, which may be a novel study to investigate the functional impact of age and Alzheimer's related changes in locus coeruleus. And I repeated it like show you like psychophysiological and neuroimaging assessments such as pupil dilation and locus coeruleus structural MRI may serve as indicators of age or individual differences in norepinephrine functioning. Lastly, I want to thank my PhD and my current mentors for fully supporting my research and thank all of my collaborators and funding agencies. Thank you.
好，那我们先感谢一下今天的讲者，就是陈翔宇的分享，然后尤其他又分享了3个有相关但是又不一样的就是研究主题这样。那我们现在就是开放给大家问任何问题，这样。那就大家可以就是现在就可以自由打开麦克风，然后可以稍微简单自我介绍一下，然后看有什么问题可以问讲者。我想除了问问题之外，这也是一个很好的机会，大家可以 network 这样子。尤其是如果刚好大家是相关领域，然后可以有学术上的交流这样子。
嗨，学姐好。我现在是在UMass Amherst 读 PhD，不过我就没有涉及到老化相关，然后是读心理与脑科学。对，然后我想问的是，如果是老年人的话，会有比较大的个体差异？
会啊，因为其实有一个 hypothesis 我们叫 neuromodulation hypothesis。它这个 hypothesis 的意思是说，这个 hypothesis 它 published by Lindenberg，它是一个我们在做 agent 的元老，然后他 propose 的这个 hypothesis 是说，当老年人不一定是老年人，就是你在就是人类老化的过程当中。老化会，比如说使你的 neurons，逐渐凋亡，或者是你的，network 的 connection 会变得更差，这样子。那这些都会反映在你的行为上面。所以在老年人身上，我们确实会看到更大的，variation，不管是在 brain 的 data 或者是在 behavior 的 data 上面。
因为我有遇过，就是几乎看不出来老化的老年人，然后也有
对。
遇过，就是对，
对，
比
那因为
较老。
年轻人上面可能比较 individual differences 可能不会比较这么大，是因为
嗯
年轻人的 brain function 都还是很完整，然后他们
嗯
有比较充足的不同的 neurotransmitter system 在 support each other 这样子。对，
嗯，
所以你可以把它想成年轻人大部分可能都达到一个 ceiling effect，就是他们的 performance 都很好。对，可是当
哦，
老化的过程当中，你的 neuron 开始有影响的，那你确实他们的 performance variation 就会比较大。那有一些研究，他们甚至去看，在我们心理系里面，我们很常会去测量 reaction time，right？就是那个你的反应时间。
对，
那我们还会看到说，如果你去看反应时间的standard deviation，它就是一个 variation 的 term 嘛。你在老年人身上，它的 standard deviation in the reaction time is larger than younger adults。
嗯
所以这个在行为上面，其实我们从我们行为，就是我们心理行为上面很常，就是最基本的一些行为指标，就已经可以看到那个 variation 这样子。嗯，
那对于延迟或是减缓老化的这个负面
哦，
效果有什么建议吗？
这个我的受试者很常问我这个问题，但我必须要说，我们文化里面有一个谚语啊："活到老，学到老"，确实就是这样。就是你一直要不断的 keep 自己在 learning，然后
嗯
explore in different activity，even it's a social activity，it's also fine。然后运动，对。那最近我的那个 curiosity study 呢，会跟另外一个博后，就是 Jenny Crawford，也是在我们实验室的另外一个博后，他的 study 主要是去看 daily life activity，所以他提供受试者呢那个 Oura Ring，就是我们现在不是有那个 Apple Watch，然后现在有一种是那个
嗯
就是戒指的那一种，它也可以去测量
嗯
他的 sleep quality daily life activity。所以我们未来会去看说，受试者他们如果日常生活当中 explore 到更多的地方，就是他们 daily life activity 更丰富的话，会不会是一种 resilience effects 在他们的 cognitive function 上面？对，
嗯，
嗯
好，谢谢学姐。
你好，我想请教一下，就是
嗯
关于你刚刚提到，其实有点 follow up 刚刚的问题，就
嗯，
是在老年人，因为听起来是你应该是用那个 clinical age，就是我们的年代年龄还是什么，anyway，然后但
哦
是不是用，
，chronological age，yeah，
对对对，但是不是用 biological age，
嗯哼，
呃，因为在一些 study 里头显现出说，我们的 DNA methylation 或者是我们其实它会直接反映出 biological age，所
嗯。
以我在猜想，老年人之所以变化那么就是 variation 比较大的原因之一，有可能是因为他没有参照到 biological age。
对，没错，
那
就是
那
这个，不好意思，你
对对对，
你说完。
然后我在想说，有没有可能就是同时测量一些不过这比较偏一些genome 或 sequencing 的部分呢，就是想说有没有可能针对某些 locus，然后去看它的 methylation pattern 来推估它大致上的 biological age，然后来做 correlation。因为我觉得呃反应迟钝、反应变化这部分的 variation，其实蛮大一部分应该也会直接被成长的环境给影响到，那会反映出呃在在在 epigenetics modification 或 biological age 的
嗯
变化上。
嗯，
对，
我觉得这是一个很好的问题。然后确实，在我们做 aging research 里面，有人在就是探讨这个问题。我们通常
嗯
是把它分成 chronological age，which is a biological age。那另外一种的话，我们会称为是 cognitive age。
嗯哼
对，那如果你有兴趣的话，你可以看那个 Dr. 陈曦，他是就是我猜他现在好像在 Stony Brook University 当 PI，然后他的实验就是去用一些 neuropsychological 的battery，像那些 behavior tests，然后去看老年人他会去换算说老年人他现在的 cognitive age 是怎样。对，那确实你会看到一个很大的 variation。有些老年人他的 cognitive age 可能就是 younger than their biological age or vice versa。嗯，然后他也会有去看说，那这样子的一个 difference 跟他的脑部的变化会不会有关系？对，
好，谢谢。
我们还有一点点时间，所以大家可以把握时间，就是问问题这样子。
或者是大家如果有，因为我算是有不同的经历我在德国念 PhD，然后现在在美国当博后。如果你们对于有博后的疑问，或者是未来想去国外念博士的疑问的话，都可以随时提问。
然后香宇自己本身之前也是拿就是留学奖学金的，所以他好像对于申请奖学金以及大家可以看到他有拿过 DAAD 的一些房地这样子，所以其实这些对于不管是学生时期还是就是像 postdoc，就是算是已经偏 semi independent 研究的人，其实这些经验都是
嗯
还蛮可以参考的，所以其实大家也都可以问这样子。然后或者是跨国经验啦，就是我跟香宇都是从德国、美
嗯，
国，其实国
我同事
家之间，对对对对，就是研究不管是氛围啊，或者是环境啊，或者是与人相处，其实都差蛮多。
对，
嗯，
我同事都说我在环游世界。
那不然我问一个好了，就是我不是做 clinical 的，我是做 mouse experiment 的，我很好奇，像你们这样 agent 研究，我相信绝对有老鼠嘛。那目前看起来，就是老鼠 model 以及人的结果，就是它的就是 correlation 的高吗？我不知道。
我觉得当然你一定会看到一些 inconsistent findings，但是目前我觉得就 previous literature 来看的话，I would say most of the evidence are consistent。Like，对，你在老鼠上面看到，因为尤其是小鼠，它们大脑的结构跟人类大脑结构长相当然不一样了，可是就是比如说 locus coeruleus 他们也有，那多巴明他们也有。所以在他们在动物 model 上面看到的一些 effects，确实在嗯人类身上还是会发现。我觉得就只是差在于那个 animal model，你可以更直接的去看说 firing rate 那种 causal result relationship，但是在人类身上的话，你也不可能就是直接插一根针在就是人类受试者身上。所以困难度确实就会比较高一些。嗯，
那通常像你们研究，就是当然你们 literature search 的时候，针对老鼠出来的结果，对你们参考性通常会很大吗？就是会
很大
OK。
很大，而且因为让我们领域的话，大部分 animal studies 他们会比我们可能走在更前端一点点。对，那我们通常也会去看，虽然我自己不是做动物的，可是我很长的时候也要去了解动物实验。然后一来是你才可以去 update 那些，因为像近几年有一个就是陆续有一些 animal studies，他们发现说 locus coeruleus 它不仅是会 release norepinephrine，它同时也会 release dopamine，所以它是 both dopamine and norepinephrine into the hippocampus region。所以，但这个的话，在我们以前的话，我们可能就会想说，哦，locus coeruleus 就只有 norepinephrine，但其实不是，就是在小鼠的那个 model 上面就已经看到它同时会 release 两种不同的 neurotransmitter 带到 hippocampus，就是 memory region 上面。对， 那所以在我们人类，尤其是在我们实验室的话，因为我目前的 mentor 他的 project 他有 dopamine related project，那我是在 norepinephrine related project 里面。可是我们所有 data 像 FNT，刚才我 present 给你的，我们除了去看 norepinephrine 之外，也可以去看 dopamine，因为它们的源头是一样的。对，所以在这种情况之下的话，我们就可以去看，就是两边 dopamine 跟 norepinephrine at the same time，呀。
好的，谢谢。那稍微打岔一下，所以我刚才有在 chat box 放了几个 link，然后就是还是欢迎大家可以 follow Tyra，如果未来有相关里面的 talk，大家如果有兴趣可以报名参加。同时，我们这一场活动我也有贴一个就是 post event survey，那大家也可以花个几分钟帮我们填写一下，因为你们的就是回馈对我们蛮重要。那如果觉得今天的 talk 还不错，或者有什么话想要跟讲者说的，也可以留下，然后我们会帮你传达给讲者，这样子。对。
嗯
OK，那或许我可以现在先stop recording。OK。
，OK。